Hello, I am Susan Grew, the Director of Programs for the Newport Beach Public Library Foundation, and I welcome you tonight to our last Medicine in Our Backyard lecture program for the year. But don't worry, we will start again in September, and UC Irvine Health has already given us a wonderful slate of doctors, and we will be ready to go in September. We would like to, before we get started, I would like to thank Ike and Ginny Kempler and UC Irvine Health for the sponsorship of this wonderful series. And also, again, to thank UCI Health for sharing their wonderful doctors with us, and for all of the doctors to take time out of their busy schedules to be here tonight to share their expertise with us. Don't forget to visit. Kamala and Michelle are in the back, and they have information not only on tonight's speakers, which I think most of you collected, but also additional information on stem cell research and on the Gavin Herbert Eye Institute. And they'll be here after the lecture, and they can answer questions and help you get the doctor's contact information. I have just a quick bit to say about the Newport Beach Public Library Foundation. It is a membership-driven organization, and what that means is we don't receive any funding from the city. Our whole goal is to support this wonderful Newport Beach Public Library and to provide stimulating programs such as tonight's program to you, the community. If you're not already a foundation member, I ask you to please consider joining tonight. In the back of the room, you will find membership applications, and they're embedded in our beautiful bookmark newsletter that has our last Witty Speaker of the Year, National Book Award winner Masha Gessen on the cover. And a few quick reminders. This evening's program is being video recorded and will be available on the Newport Beach Public Library Foundation's website. Usually takes about a week to get up on the website, but please go to mbplfoundation.org and look under the Medicine in Our Backyard lecture series page, and you will find this lecture and all of the lectures for the past year. Also, a gentle reminder, but a firm reminder, I'm going to bring out my mom voice here. Please silence your cell phones. I'm not asking you to turn them off. I understand sometimes we need to be reached, and you never know, but please silence them. It is such a gesture of respect to our lecturers to have a silent and attentive audience. And then our final reminder is this lecture has a built-in long ending, which gives you the opportunity to ask questions of the doctors. Yes, thank you. That's good. Let's get it all out now. We build this long question and answer session into the program because we know you're all here to hear not only the doctor's lectures, but to also ask them questions that you've brought here tonight to get answered. And then finally, after the formal Q&A session, we invite you to come and meet both of the doctors face-to-face after the program. In a minute, I'm going to bring up our wonderful and dedicated chairman of the board, Dorothy Larson, to introduce Dr. Bota. But before I do that, I'm going to say a quick word about our other fabulous speaker tonight, Dr. Henry Clausen. Dr. Clausen serves as the research director for the Retinal Regeneration program at UCI Health as part of the Gavin Herbert Eye Institute, and he is affiliated with the Sue and Bill Gross Stem Cell Research Center. Dr. Clausen focuses on regenerating damaged retinal tissue to restore sight to those suffering from retinitis pigmentosa and macular degeneration. We welcome Dr. Clausen here to the Newport Beach Library. But before we do that, I would like to bring up my board chair, Dorothy Larson, to introduce Dr. Bota.
Good evening. This may seem a little bit redundant or unnecessary. My name's Dorothy Larson. I am the chairman of the board, proud to be, of the Newport Beach Public Library Foundation, and I've always been really proud of the great depth and diversity of programs that we have here. But I was so excited when I found out that Dr. Bota was going to be here to talk to you about clinical trials. It is such an important topic. she is a neuro-oncologist at UCI Medical Center in Orange, and she was the lead doctor in the clinical trial that my husband participated in a couple of years ago when he was diagnosed with a glioblastoma. So from a personal perspective, I can tell you what I mentioned to her tonight. I was told by a physician friend, don't go to the nearest hospital or the nearest emergency room when we figured out there was really a problem. Go to the hospital that's going to be able to take the best care of him. So I took a few minutes before we left and did as much research as I possibly could, and that's how we ended up with Dr. Bota at UCI Medical Center in Orange, and they do amazing work. They are all skilled physicians that just have an amazing ability to do research, and at the same time that they're doing really important, cutting-edge research that is very, very technical, they are caring about the whole person, the whole patient, the patient's family, the process. So that was one of the more amazing things that I experienced, and wanted to share that with you as I bring up Dr. Daniela Bota. I could read a long list of her accomplishments, but I promised that I wouldn't. But you can Google her and get a list of research publications that she's done. With that, Dr. Daniela Bota, thank you.
Perfect. Thank you so much for being here tonight. Tonight, I would like to share with you some of the ideas that are powering our clinical research enterprise. I'm here to talk to you in multiple levels and multiple capacities. Maybe the most important things that I'm doing right now administratively is that I'm the Associate Dean for Clinical Research for University of California, Irvine School of Medicine. And with that, I'm trying, and we are all trying together to bring the most innovative, the latest stages of research to our clinical enterprise. I'm a neuro-oncologist. I'm also a stem cell biologist. So what you're going to hear tonight is going to be a very short and very fast, going through what clinical trials are, how can we get them from the idea of a clinical trial to our patients, how can we help people? How can we benefit people? But also, and most important, how to be an informed consumer of clinical trials. What is really a clinical trial versus what is something that's masquerading as a clinical trial? And how can you fully understand and fully protect yourself as well as your loved ones and participate on the science that will move us forward? So, I don't know if everybody knew or if this was special. I really thought that Dorothy knew and this was very special. But yesterday, actually, it was our celebration. May 20 is the International Clinical Trial Day. And what we are celebrating on that day, it's one of the very first studies. And that is the study of Dr. James Lind. And he experimented first time, how do you treat scurry? And, he gave some people vitamin C, and those people did very well. So we are celebrating International Clinical Trial Day. We are just one day after, so happy belated anniversary to all of us. And why would we do clinical trials? What is our mission? Our mission to do clinical trial is stemming from those important points. We want to discover leading-edge treatments in the lab. A lot of us are having research groups where we experiment in cells, then we move on animal systems. We are trying to get all the information that we can. But after all that information is obtained, the next very important step is taking that and being able to translate. And translate is the key word, because we want to move everything that we learn in the lab and be able to bring in active clinical trials. And all of us are very blessed. We live in California, and we live in a place where science discoveries take place every day. And there's so many research institutions and so many private enterprises. And we are here to help all that collective intelligence become involved into bringing new treatments and new cure to the people around us. When we think about clinical trial development and clinical research development in general, we have to remember that there is a very clear structure, and the reason for which I am insisting on this very clear structure is because it's true. Clinical research takes time and takes money, and it takes also political influence and political power. So what I'm talking about is we have to start with basic research on which we try to look at safety, utility, and how to make it into an industrial-like form, a treatment, a drug, a device. We do a prototype design, we do a preclinical development in cells and animals. We do clinical development, again, in human and animal testing, and then all this information going now over years, including our manufacturing, including our safety, including our specifications, so those things are safe, finally get FDA filed, FDA approved, and in mass production. So you can understand that all the dimensions on vertical and horizontal will involve very large teams and will take time and effort. So when we talk about clinical trials, it's also very important that we all talk the same language, because what one person calls a trial, another person might call something else. And clinical trials, the biggest misunderstanding is that majority of the people will think that clinical trials are basically treatment trials. You are trying to find out if a treatment, new or old, a combination of treatments, will work. But we also have prevention trials. We are trying to prevent heart disease. We are trying to prevent Alzheimer's dementia. A lot of work is done not on treating conditions that we already have, but on the equally important enterprise of preventing future conditions. We have diagnostic trials where we are developing new, maybe radiological modalities or blood tests. We have screening trials where we try to identify who are the people that are most commonly developing a disease. Can we predict based on genetic factors or lifestyle conditions who are the people on which we have to concentrate? And then we have quality of life trials. And this is so important because we always rush for cure. I'm a cancer doctor. We want to cure cancer. But the question is, while we are curing the cancer, what kind of damage are we doing to the normal body? That the patient that is cured or in remission has a very poor quality of life because we destroyed other essential parts. So all of this together is what defines clinical trials. Another very important language that we have to learn is understanding what it means that you are in a phase 1, phase 2, phase 3, or a phase 4. Because many of our patients, this is the first decision that they have to make. They have to ask themselves and ask their families what is their level of risk they want to take. Remember, all of those are experiments. There is a great hope that some of those will be transformed into cures, but they are experiments. We don't know if they're going to be successful. And where in the level of discovery are you comfortable being? Because when we talk about a phase 1 study, this is a study that we are just trying to find out what's the right dose. We are starting from a dose that's lower and we hope it's safe. We treat a few patients. If that's tolerated, we move to the next dose and we keep going up and up until we find a dose that many times is not well tolerated, and then we go a little bit lower down. So this is a dose finding study. When we go on the phase 2, the safety part has already been at least partially explored, and we are trying to find more definitions of safety and get preliminary evaluation of this treatment being effective. On phase 1, very rarely we have evidences of things being effective. And then we move to the phase 3 trials. The phase 3 trials are those very large studies that you hear. They involve hundreds and thousands of patients. Many times there is a control group. Many times there is a randomization, which means that though the treatment that's offered, it's maybe very advanced, very promising, there is a chance that the patients will be randomized to a control group. So half of the patients or a third of the patients, depending on how the study is designed, will receive the standard of care treatment. They are not going to receive the new experimental treatment. The phase 4 studies are post-marketing. The drugs are now approved, but the drugs that are approved are studied maybe for 2 years, 3 years, 5 years. So now that the drugs are approved, we are embarking ourselves in the 10 years and 20 years data so we can understand the long-term safety and the long-term efficacy of those treatments. I hope that you guys will gonna be shocked, because I am every time that I show this. This is the total global spending on research and development. So this is money that states and pharmaceutical companies will invest in drug development, and we are getting close to $200 billion a year. And this has been ongoing. This is 2008 to 2022, and you can see that the number is now higher with 50%. This is the number of registered clinical trials, officially registered, and if you look from 2000, when we had about 5,000 clinical trials, now we have 251,000 clinical trials as of last year. So the wealth of clinical research is enormous, and that is why everybody, in my opinion, needs a primer to be able to navigate this type of information. So what are we researching? Most of the times we are researching infectious diseases. Yes, HIV, we still have to discover things, malaria, the Gates Foundation. But for the purpose today, very importantly, we are spending almost the same amount of time and of money on oncology, neurology, cardiovascular diseases, disorders of the civilized world, of the advanced economical states, and this is where a lot of the monies of industry go, because this is where they hope that the return on investment will arrive. I hope that many of you are familiar with the 21st Century Cure Act. This was a law that was approved in 2016, and the purpose of this law was to accelerate the development of drugs and devices and to bring new information for the patients faster and more efficient. Why was this important? You guys know FDA is tasked to provide for our safety, what is happening is that there's many times the public is perceiving the FDA as being very conservative and is looking at the fact that usually it takes 10 years, maybe even 20 years from the time that a drug is ready to leave the lab or a device or a cellular therapy until that modality gets to the patients in the clinic. So the public has asked for a shortening of that time, but still was keeping the rules for safety, because we all want to be safe. I think some of the people my generation and even older remember the thalidomide story. How many babies got harmed with a drug that was supposed to be a benign nausea and headache treatment for pregnant ladies. So, with this law, what we'll talk about today more is the Regenerative Medicine Advanced Therapy and the Breakthrough Designation Program. It was also the creation of an Oncology Center of Excellence that provides patient-centered decision-making. The proposal that we are all paying, it's $500 million over 9 years, and is reporting directly to the Congress from the FDA. So, regenerative medicine is what we are talking about today, the power of harnessing stem cells and using them to heal our bodies. And when we talk about that, what's happening with this law is that we have a number of methods through which those treatments can go in the clinic faster. We have a fast-track designation, which is an early type of approval. If the clinical trial is promising, we have a breakthrough therapy designation, which helps the FDA to identify early things that are very promising and move them faster to development. We have the RMAT designation, which is very similar with the breakthrough therapy designation, but it's specific for stem cells. It doesn't include oncological drugs or devices. And then we have the priority review designation, where all of those, once the final data are collected, the data can be reviewed in much faster way, and treatments can be brought faster to the patients as FDA-approved treatments. So, what is the RMAT designation? The RMAT designation means that a product is eligible for development and review, and this will increase the meeting opportunities with the FDA, will increase the early interaction, and will accelerate the approval. Everything that we are developing for drugs or therapeutic agents will be eligible if we'll have a therapy that's defined as a therapy for cellular, tissue products, or a combination. It will treat, modify, or cure a serious and life-threatening disease. And I think this is important to notice. Those advances in the FDA processing are for the diseases they need the most. Those are the life-threatening disease. Those are the Alzheimer and the Parkinson and the blindness and the cancer. Those are not things to try to get another blood pressure medication on the market. And it required that we have strong clinical data. Even if it's a big need, in absence of clinical data, you cannot go on this pathway. What I wanted to show you is also a list of the treatments that already have an RMAT designation, so they're accelerated through the trials. I know that some of those treatments will be extensively discussed in the second part of this lecture. But the reason for which I'm showing you the list is also because a number of those treatments, in addition to the retinitis pigmentosa that we're going to hear a lot about in the second part, are already in clinical trials at UC Irvine in the alpha stem cell clinic. So we are trying already to bring all of those potential treatments to the patients in Southern California. I'm going to change a little bit the topic. Of course, I have to talk at least a few minutes about glioblastoma. And this is a very aggressive brain cancer. The survival, unfortunately, is still around a year and a half. The tumor can double in size every 2 weeks, and unfortunately, we have only 2 approved therapies. The part that we are focusing on in my lab is the idea that the normal cells of the brain, the neural stem cells, are choosing a pathway of development on which they are becoming cancer stem cells, and those cancer stem cells keep repopulating this very high-growing, fast-growing cancer. We started our work by screening a number of compounds that seem to be specific for those cells, and we ended up with a natural compound that it can be found on the coast of Bahamas. It's called marizumab. It's an antibiotic produced by this nice, cute Salinostora tropica microorganism. And this drug, it's able to go in the brain, and it has great promise in treating cancer. So the way in which we do development is that we took the drug, we proved that it works in cells, and then we had little avatar mice that were carrying human tumors. And we were able to keep those mice living longer and longer, surviving more than double than the time that the mice that were carrying the same tumors but did not get our drug. With those data, we went to the FDA together with the company that was sponsoring us, and we have obtained an orphan drug designation, and we opened the first glioblastoma study in May 2016. This was the regular phase 1 study on which we tried to find the best dose to treat, and then we took that dose and we studied in an additional 24 patients. This was one of our patients on which this very large tumor on the effect of the drug basically went on the remission for 2.5 years. The median survival on this stage of disease is usually 6 months. We are very happy to note that in many subgroups of the patients on which the usually expected survival at 9 months was 12%, we were going to 50%. This was very promising, so from the phase 1-2, we went to a phase 3, which is taking place right now, very large international study. The reason for which I'm describing this is because I want you to understand what are the steps from the lab to the phase 3 study and the FDA approval. We are going back to the CERM mission, which is to accelerate stem cell treatments to patients with unmet medical needs. I'm sure that you are all familiar with CERM. It is funded by all our money. That was a proposition that was passed, which funds this institute. And at UC Irvine, we have one of the 6 alpha stem cell clinics that are funded by the state of California. And you can see here the number of diseases that we are treating and how we are promoting them in the clinical trials. And if you're looking for us, this is how our wall looks like. But those are the active products that are right now in clinical trials at UC Irvine based on the stem cell therapy. So you'll see a number of diseases from stroke to traumatic brain injury to kidney failure to ALS. This is Lou Gehrig disease, of course, glioblastoma, burns, and aging. So wide array and phases from phase 1 to phase 3. We're also doing the research for Huntington disease And this is yet again another glioblastoma study. Diabetes, bone grafts, spinal cord injury, the study that I was telling you that's getting very close to RMAT approval. Different studies now for hip fracture. So, I will spend just 5 minutes to talk about something completely different that you have to be aware of, which is the stem cell tourism. When we think about stem cell tourism, I was having this wonderful discussion with Dorothy at dinner, and Dorothy said, that's what's happening in other countries. And I said, no, this is what's happening in Southern California. This is what's happening near us, where people, clinics are listing conditions as Alzheimer's, Parkinson, multiple sclerosis, diabetes, and cancer. They're charging patients thousands of dollars, $50,000, $60,000, providing them with therapies that are not FDA approved and without scientific evidence obtained from clinical trials. This is, as of now, an article just published in Cell Stem Cell, very important journal that we use. The places where we have stem cell clinics that are offering stem cell treatments for all those disorders for which we do not have an FDA approval to use stem cell therapies yet, because the clinical trials still need the years that I mentioned to you, even though we're accelerating things. Why I'm talking about that, I usually do not try to go on things that are controversial, is because this article in the New York Times in May 9, 2018, on which the FDA got court orders to stop 2 clinics, one of them from Southern California, California Stem Cell Treatment Centers, from receiving unapproved stem cell treatment. This was stemming from 3 cases of patients that got material extracted by fat. They were told that they are adipose stem cells injected in their eye. And those 3 patients, all of them developed blindness. In 2015, they were losing their vision, but now they developed acute blindness after this transplant. There are some arrests made, and this is a very cautionary tale that you cannot hurry science. You have to go through the right steps and the right regulatory process on all that big table that I showed you the first time, that you have to do safety, efficacy, do the right animal models, and be careful, because sometimes things that are too good to be true, they're actually too good to be true. So, 2016, there were the allegation about the patients blinded in Florida. 2017, FDA issued a letter. In 2018, the California made the disclosure law mandatory that the patients will be told what is approved and what's not. And in North Dakota, actually, is the first state that now it's regulating stem cell clinics. Our law requires the warning, requires a statement to be handled to the customer separately before the treatment begins, and it helps all of us to understand that we are taking a risk if we are going toward that pathway of care. So, when somebody offers anybody a clinical trial or a procedure that's not FDA approved, and it should be a clinical trial, because I hope I convinced you that everything that's not FDA approved, it's a trial of some sort, what questions should you do? What should you think about? You should educate yourself on the specific treatment, try to know as much as you can, assure that the treatments are an informed consent, ask for a written copy, and have the opportunity to ask all the questions that you need and have them answered appropriately. You want the study to be approved for the FDA, you want to see what was published, you want to know that you have an IRB protecting your rights, and you also want to know how much money, if any, the investigator has involved in that clinical trial, because conflicts of interest are very big in politics, but they're equally big in medicine. So, with that, I want to thank you for listening to us, reminding you that we are in your backyard, and I'm happy to take questions after Dr. Klassen finishes his presentation.
Okay, I'm glad I can still figure that out. I'm hopelessly on the Mac now, so Bill Gates has me running in circles. Okay, well, I'd like to thank the organizers and thank Dr. Bota for a great talk that kind of provides a nice introduction to what I'm going to do. So instead of a big overview of clinical trials and translational medicine and regenerative medicine, I'm going to concentrate on this one trial that involves our work with progenitor cells for treatment of blinding diseases, particularly something called retinitis pigmentosa. First off, I direct you to my disclosure statement. I do have a financial interest in what I'm going to talk about. And then I'm going to give you another caution, and that is there's a lot of technical stuff coming, but I'm going to try and be your translator so that hopefully you don't have to get lost in the morass of detail here. And with that, let's leap right into it. So the first thing I want to do is talk a little bit about retinal degeneration. What is it we're talking about here? And so that topic covers multiple different disease conditions, and I give you two here for an examples. One's the retinitis pigmentosa you keep hearing about, and the other is macular degeneration. Now, I picked these two for a reason. One is because RP is particularly severe, and that's why we're really interested in going after that as our initial clinical target. AMD is much more common, and so you may have heard about that. A little more detail is that when the doctor examines these people with these two conditions, it's really easy to tell them apart as an ophthalmologist. And that is because in RP you have these pigmented flecks all over the retina. That's what you're seeing here. That's a view of the retina through the pupil looking back there. You see these pigment flecks, and that gives the name retinitis pigmentosa. And those flecks are not supposed to be there, and that's what gives this disease its characteristics. It's part of the pathology of what's going on. But interestingly, the central part of vision, the fovea, the macula, that part of the retina is spared until late in the disease. So RP involves the whole retina, but seems to spare the central bit, at least for a while. AMD is like almost the opposite. Instead of black pigmented flecks all over the retina, you have these white kind of nodule-like things, these white blobs, and they're centered right in the middle of the retina. They're in the macula, like the name implies. So it's the macula that's degenerating, not the periphery. And that gives rise to what the patients experience. In RP, it starts out as night blindness. It proceeds to tunnel vision, and then that tunnel vision just continues to constrict as the central vision is lost as well. And it can result in complete visual disability. AMD starts right in the center of your vision, so it interferes with ability to recognize faces, reading, driving, and all these great things. The silver lining, if there is one, is that the periphery is generally spared in this disease. So with that, you see that you can almost see that these seem like opposite conditions, and yet there's this commonality. There's this fundamental problem at the microscopic level, and that is that the photoreceptors, the cells that you've heard of called rods and cones, these are the cells that actually detect light at the very beginning of the visual process. Those are the cells that are being lost in both cases. Now, there may be more going on. I'm not trying to oversimplify, but this is the critical element. When you lose your rods and cones, you can no longer detect light wherever those cells are missing. So it's like dropping out pixels on your screen. You lose enough pixels and you've got problems. So what are you going to do about this? Well, that's the challenge that faced us in my lab. It's great to want to treat it, but, start scratching your head. What are you going to do about it? Because these are relentless degenerative processes. Well, in the age of stem cells, the possibility arose that perhaps we could replace the rods and cones with the stem cells. The stem cells could come in there and make new photoreceptors. Wouldn't that be great? And that would be great. Another possibility is neuroprotection, and that means that you're using your cells to have a positive influence on the existing host photoreceptors. Those cells that are already there, you're trying to revitalize them. So the neuroprotection means it's more like a medicinal effect on the existing cells. And we've looked into both possibilities, and in the end, we settled on the idea of this neurotrophic rescue. It's not that cell replacement isn't exciting, it certainly is, but we think in terms of getting into the clinic, this neuroprotective strategy could move along a lot quicker and start benefiting patients in the earliest possible time frame. Then I just want to give you a little rationale for why that is. So remember, there's rods and cones in the retina. The rods are there to detect very dim light, moonless night, no street lights. So the rods really function under dark adapted conditions. You wake up in your bedroom and the electricity's off in the middle of the night. That's when your rods need to function. So the rods are the cells that express the mutant genes in retinitis pigmentosa. It's just the rods. So those are the cells that degenerate first, and that's why the people get night blindness as their initial condition. Unfortunately, the cones then start to degenerate as a knock-on effect. So they're not expressing the mutant gene, and yet they die. And when they die, everything goes with them. So all your color vision, face recognition, high definition vision, it all goes. So if we could just get in there and rescue these cones that don't even express the mutant gene, then that's our rationale. That's kind of our cognitive traction to try to get some leverage on this problem. Just interfere with this degenerative process, rescue these cones. Okay, sounds good, but how do you do it? Well, that's where these retinal progenitor cells come into play. So this is a project I started 20 years ago at Chalk up the road a ways. And this is our hero here, a retinal progenitor cell, one of them seen at very high power. So now it gets a little technical. I'll skip over this. I call them progenitor cells because they're technically not a stem cell, but they're a lot like a stem cell. So from a patient standpoint, okay, they're a stem cell. But from a scientist standpoint, you can't really say that. So just be aware there are some differences at the level of the aficionado. But regardless, those cells have low immunogenicity, and there's a lot of good things about them. And I'll cover some of those topics as we go here. Again, we are going to focus on RP. It's a very severe condition. There are no existing treatments that really halt the progression of that disease. So the risk-benefit type of ratio makes a lot of sense for trying new therapies in RP. And a lot of new therapies have been tried, and a lot of new therapies have failed. So that's the downside, is it's really hard to treat, obviously. It's also a rare disease, but that gives you certain benefits from the FDA in the modern era because of the orphan designation. So that has a good side as well as a downside. So we did a lot of preliminary work. That work involved, of course, the usual suspects, and by that I mean rats and mice. But we also did some work with larger animals, and here I reference this green pig. That's a whole nother topic, but for the old timers, I wanted to, show them one of those just for the memories. That was really a lot of fun. They really are green, and they glow in the dark, but only when you shine UV light on them. Anyway, so after the animal work was under control, we wanted to pursue the bench to bedside element of this. That means, as Daniela was saying, you get into a translational pathway. So you're not just, the wizard dabbling with your, ideas in the laboratory. Now you have to come up with a plan. how do I take this animal data and get to the point where it's reasonable to go into a human being, and the FDA is going to agree with that. Okay, so that's the translational pathway. It's like, party's over, let's get serious. And so what you have to do is you come to the FDA with a preclinical package, and this is what the package looks like. Obviously, it's much thicker than this. This is just a brief overview, if you will. It's actually a massive amount of work. We estimate it as something like 5 to 10 different PhD theses all kind of stapled together, just to give you a sense. But anyway, there are these different components. The first one is all about the cells. So you have to provide a lot of material demonstrating why you think these cells are safe to use in humans, because that's the actual product. That's what's going in there. So the FDA rightly cares a huge amount about that. Remember in these, in the stem cell tourism, they skipped that part, Here you go. But in fact, it's good to document the history of the cells, the provenance, and everything about them, and every way you've tested them to assure people that there are no contaminating organisms, microorganisms and whatnot in your product that could do serious harm to the patient. Imagine there's HIV in there, just for starters. Patient's not going to be very happy when they find out that you didn't test for that, right? So that's just an example. Then toxicology. These are cells. These are progenitor cells. It's a newfangled type of treatment. We don't have a lot of experience out there in the community with progenitor cells, do we? So you're going to want to go into your rodents. You're going to take your product that was generated for the clinic, you're going to test that again. Even though you've done it already in rodents, you're going to do it again. You're going to do it with that clinical product, that specific product. You're going to go back in these animals, and now you're going to document every last thing that happens in those animals. You're not going to just cherry pick the part you like. You're going to document everything that happened. How many of them died? Did they all live to the end of the time frame? How many time frames did you look at? Just a week? Or did you go to the life of the animal? You see what I mean? So it gets very involved. It involves hundreds of animals and millions of dollars, to be honest. So it's very expensive. And I'm talking about the whole package here. But then you got to repeat your proof of concept. So you have your clinical product, and then you're going to go back in and show that it works. Now that you've made it safe, does it still work? Did it retain the aspect of functionality? Because you're going into humans again. We want to have more evidence that you're on track here. Then, of course, you have to have your clinical protocol. You haven't treated anybody yet, but you have to say, well, who is it you're going to treat, and how are you going to follow them, and things like that. So it all has to get laid out. And then you apply. So we did that end of 2014. And now just a quick look at each of these components. That's our product. We're very proud of that. It looks really spiffy all there in the nice little vials. Those are cryo vials, so the product, because it's a living cell, they have to be stored in the deep freeze, so they're in the liquid nitrogen tanks. That's where they hang out until we need them. Then they come out, and then we thaw them again for use in the patient. And this is just to give you a flavor of the immense amount of documentation involved. This is testing in the animals. Here you see the human progenitor product. Inside the eye, this is a mouse eye. And it's injected through the side of the eye, just like we're going to do in the patients. And the cells clump together in this is the vitreous gel area back here behind the lens. This is a lens in a mouse. It looks really big. But that's where it is. And then this multi-layered structure here, that's the retina. That's the actual retina. And you can see that looks nice and layered, and it's not disorganized. So there's no local toxic effects. The cells are well tolerated. They're not making a tumor. Everything looks fine. Meanwhile, you also have to look at the rest of the mouse and make sure that, the kidney functions okay and so on and so forth. Another thing you want to do is ask the question, where did the product end up? Okay, so you put it here, where did it go? From this, it looks like it stayed right there. And in fact, that's exactly what happens. We looked at many different tissues using a technique that would detect theoretically one single human cell and never found a single cell outside of the eye. So that provides another reassurance of safety that these cells don't just get up and start cruising around, getting into trouble. And then this is the part where we want to demonstrate that the cells still function after they've been generated as a clinical product. So this gives you a little sense of what's going on. This is the mechanism of action. So over here on the far left, we have a section through an eye of a rat. The arch structure up here with the layers, that's the retina. And this colorful blob here is the graft of progenitor cells. So they were injected as a slurry of cells, and very quickly they just kind of grab onto each other and form this nugget we call neurosphere. And that neurosphere just hangs out in the vitreous. So it's just sitting there in the jelly like it's a petri dish, if you will. And from there, it secretes various factors, protein factors, that diffuse into the retina, and that's the drug. So you can literally think of this graft as a drug factory that's sitting in the vitreous, a nice little space to hang out, and it's just spewing out this drug product constantly. And that's basically what's going on. So what is the impact of this treatment? This is a rat retina that's been splayed out. So normally you can appreciate a retina's got a bowl-like shape because it's on the back of the eye. So it's like a hemisphere. So to analyze it, we have to kind of flatten that out. And so it gets sliced in kind of like a radish maybe, and then laid down flat. And then we can zoom in with a microscope, and using special antibodies that label the rods and cones, we can look at what's going on. So if we take this retina and we label it with a green fluorescent antibody for a middle wavelength cone, and then we use a different red antibody that labels the rod, the rhodopsin that's in the rod, and has a nice red tag on it, then we can zoom in at high power and see what's going on. And so what you see here are two different animals. This is the retina from two different animals. Both of them are going blind. They both have retinal degeneration. In this case, the animal was not treated. And you're looking for red and green and you don't see much, right? Because it's all gone. The rods and cones are gone. Whereas in this case, you see the red and the green. It's more obvious when the lights are lower, but then you'd all be asleep. So trust me, there's red and there's green there. And those are individual photoreceptors. And the green ones are cones and the red ones are rods. And so the point is, this is the retina you'd rather have. Now, it's one thing to show that the rods and cones are still there, but we need to demonstrate that these are functional rods and cones. They're not just there for decoration, okay? Because what the patient really cares about is whether they're functioning. So we have two different tests we perform on these animals. The top one is a head tracking response. It's a behavior. The way for you guys to think about it is when you go to the eye doctor, you have to read the eye chart. But that's because you can follow directions and you can read. Rodents don't follow directions and they can't read, right? So we use this funny head tracking device that we can use to approximate their visual acuity, and this is the readout. And in this graph, the higher the bar, the better the animal can see. And this green one is the cell-treated animal, and these are the control animals. So you see that they could see somewhat better with the cells. Now, the more definitive test is the ERG, that's electroretinogram. Now, most of you probably haven't had that performed on you, but you've probably had a cardiogram at some point, especially when you get to be my age. And so, you just put a few electrodes on there and it measures electrical activity as the muscle goes through its cycle. Well, you can measure electrical activity from the retina, and all you have to do is put some surface electrodes on the eye and then flash a light, and you see the response of the retinal circuitry without having to stick an electrode in or anything. It's just like surface electrodes. So we can do this on little animals too. And again, here's our control animals, and you see that we have a higher bar here, means that it's a better performance when we treat with cells. And in this case, we have like a gradation of different cell concentrations. So we're giving a higher dose as we go this way, and you see this kind of trend upward. So that's our functional assessment. And in fact, ERG is a functional assessment that's used to diagnose RP in human beings. So there's a direct relevance there. So then the overview of what we're planning to do in the patients. So we're going to take this retinal product from these vials. It's allogeneic. That means it's not from the patient. It's from a donation from an unrelated source. We're going to put it into people with RP. We're going to deliver it by an intraocular injection. So it's going to be injected into the vitreous, remember, into the gel of the eye, just like you saw in the mice. We perform that under numbing drops, so the patient isn't put to sleep. They just get a numb eye, so it's really an office procedure. And importantly, we are not going to immunosuppress the people, even though these cells come from an unrelated donor. And just to reemphasize, we're trying to use these cells to provide a neurotrophic effect. We're not using them to make new photoreceptors. Okay, now we get into the business. So first off, to do this translational project, you need the funding to drive it forward, or you won't get too far. So we've partnered with CIRM with the funding for this. So again, that's the California Institute of Regenerative Medicine. That's the Stem Cell Institute. And so we got this large grant that covered both the preclinical package I just described and the phase 1/2A trial that I'm about to describe. So it got us all the way through preclinical and early clinical, and that's why there's a lot of money involved. We also partnered with a group at the NIH that's interested in rare diseases, and remember, RP is a rare disease. And then we founded a startup company, JSite. JSite's just a few blocks away from here. So when you're driving by, wave. And JSite started off with humble beginnings, spin-off of UCI. But its first task was to license the intellectual property. That means patents. So the patents all belong to University of California. So JSite would license that to further develop it into a commercial product. At this point, JSite's involved in many more aspects of this, certainly grown quite a bit. In terms of the FDA, you heard earlier that there's a lot of interactions with the FDA. We did get the orphan designation because RP's rare. We had an early meeting, we had a later meeting. We keep having meetings. And so the interaction with the FDA is an ongoing type of thing. And then there's the clinical trial. So we manufactured our clinical product up at UC Davis, just so you know. So that's where these cells stay in the liquid nitrogen until they're ready to come down for use in a patient. And that's when we prepare them in the building you see here. That's the Sue and Bill Gross Stem Cell Center at UC Irvine. We have a special cell preparation facility developed inside of that building. So we take the cells out of the deep freeze and we get them all ready. And Jing here, who's back there, so hi Jing. She prepares them for the patient, and she's the one normally taking them over. I was doing this the first time, probably just for the photograph. You can see how relaxed I felt at the time. And so that's where we are. So then we had 2 clinical sites to begin with, and we continue to have those 2 sites. One is right across the parking lot here at the Gavin Herbert Eye Institute, and the other one's up in LA. It's a private ophthalmology practice run by David Boyer and very famous for doing cutting-edge trials, so a really great resource. Both of them are. This is up at RVA in Los Angeles. This was the very first patient that we dosed in this trial. The patient's just in the blank area of the screen, but I was catching the interest in the hallway. People were gathered to see this unusual event of a stem cell treatment being tested out in somebody. We started out with 2 different cohorts of patients. So the first cohort of patient would be the people who were the most severely affected, and then we moved into people who were somewhat better seeing patients, but realistically they were still severely impaired by their disease. The enrollment went very well. We had 4 different dose levels, and we completed enrollment in July of 2016. All patients made it through the whole trial. We followed them for a year and finished the trial in August of 2017. So that's really great. In terms of safety, the safety profile was good. There's always something that goes on. Most of the things that we saw recorded related to this treatment really were attributable just to the fact that there was an injection in the eye. In other words, bloodshot eye, a little bit of eye pain for a while, this and that. So it was pretty much just the injection. There might have been a little inflammation on top of that, which was readily treated with the usual steroids and so on. And importantly, we didn't see the types of things that people might be concerned about with this kind of treatment. Anytime you're using a cell-based treatment, you're going to be concerned if the cells are going to grow inside the patient. That was not an issue. Au contraire, it goes more the other way. And then there's something called epiretinal membrane. You got to be concerned if these cells spread out on the retina and pull it off, make a detachment. Didn't see that either. And then, of course, immune rejection was a big concern because we didn't immunosuppress the patients and we didn't match the cells, and yet they were well tolerated, so immune rejection was not an issue. In terms of efficacy, I just want to caution, this was not an efficacy trial, it was a safety trial, but we use visual acuity as our primary measure, but that's a very high bar to get over. Nevertheless, we did see visual improvement in these patients as a group versus the untreated eye. We also saw evidence of improved vision versus the baseline in the same eye. And if we compare the vision in the treated eye to what we would expect it to be based on historical progression of the disease, again, we saw evidence of improvement in vision. And this improvement was present at both 6 months and the final time point of 12 months. So all told, we were very gratified to see this signal of improved vision in these patients. Extension trial. So people have all finished the first trial. All of those patients were entered into the extension trial, and they were offered an opportunity to be dosed in the other eye, and most of them took us up on that offer, so we've dosed people in the other eye, and that was also well tolerated. You heard about the RMAT designation. We got that designation from the FDA, so we're looking forward to working with the FDA to try and accelerate this project to get it into people as soon as possible. And then we've moved on from the first trial. We're now in the phase 2B trial. This is a larger trial. It's dedicated to looking for evidence of efficacy. So we saw some suggestion there, but now we want to prove it statistically. So we're going to have treatment group in which you get the cell, but we also have a treatment condition, so to speak, where you do not get the cell. And both the patients and the providers are masked in terms of who got what, so that in the end we can do a statistical analysis and demonstrate whether treatment is better than no treatment or sham treatment. And enrollment on that's going good. And then just a quick shout out that we think this has application in other diseases. We picked RP because of the risk-benefit ratio and so on, but it's not that this treatment was tailored to RP. We think it has broad applicability in conditions in which retinal neurons are undergoing degeneration. And that includes diseases involving retinal ganglion cells in the optic nerve. So glaucoma and so on, optic nerve neuropathy and so on might be amenable to this. And then just big thanks to everybody involved, particularly Jing, who's here tonight. Hi, Jing. And that's the Stem Cell Center in our team, and that's the Eye Center you're all familiar with, right across the parking lot. And here's our group at J-Site. I should include a picture of them as well. All right, thank you very much.
Thank you, Dr. Claessens and Dr. Bota. I invite you to both come up and collect some questions from the audience. So, audience, if you want to raise your hand, and if you feel comfortable standing, that will help you project. The doctors will repeat your question. And go ahead, take it away.
Let's start from this corner. I think that you wanted to ask the first question. Please go ahead.
Ours? Okay. So we used a cutoff of 18 years, people who can legally give their own consent. And from there, we didn't actually put a limit. So it was anybody 18 or older.
What type of food would you suggest to be a form of preventive for macular degeneration? If you don't have it you maybe be eating some type of food that would...
Yeah, people usually recommend leafy green vegetables and colored squashes and things like that. So vegetables with pigment is probably a good idea.
And I'm going to remind the doctors to repeat the question, please. Thank you.
Oh, sorry.
I just wondered what criteria you have to be in the study.
To be in the study, the RP study?
Yes.
So first off, you have to have RP. We heard about the age requirement. There's certain disqualifications related to severe systemic diseases. In other words, you don't want the data to be made complicated by people dying of other causes that was predictable. So if you had terminal cancer, you're not eligible because that's just going to mess up the results. And then, so then other eye conditions could be a complicating factor, although if you've had, for instance, your lens replaced with an intraocular lens, that's okay. So it just depends. You got to get into the detail there. There's also criteria related to what stage of the disease you are, and that relates specifically to what we're trying to achieve. So balancing also the safety factor. So we're not moving into people who have 20/20 vision at this point because we want to get a stronger safety record before we put somebody with 20/20 at risk. In fact, we're looking at people in the current study from 2800 to 2080. That's our starting point.
think that was the next person.
Do you anticipate doing studies on the optic nerve anytime soon yourself, or?
So the question is, what's our anticipated trajectory on optic nerve conditions? That's undetermined right now. We're very focused on pushing through on RP. Probably next up are other degenerations. But optic nerve, it just depends on the resources available to generate a large enough team to have redundant people, because in order to start doing things in parallel, you just literally need more resources. Otherwise, it ends up being where you have to do one at a time.
Well, specifically for glaucoma, which I know is not an orphan.
Correct. I mean,
So that means it's going to be like decades or?
I hope not. I don't have decades.
Go ahead.
This question is for you, Dr. Bota. On a patient with glioblastoma that has been MGMT methylated, so did really well with the temodar radiation, and the tumor goes away, would you recommend immunotherapy before a comeback or after?
Again, this is a very important question. It's important when the tumor goes into remission for the patient to continue to be followed. For clinical trials, we do have both options. There are clinical trials on which we treat patients that have tumor progression, the tumor comes back and is growing, but there are also clinical trials on which the goal is maintenance. Many of you guys are familiar. For breast cancer, by example, for many of the patients, after the cancer is into remission, there are other drugs that are used during the maintenance period. So both of those times in the patient's treatment are important, but they have to be approached in different clinical trials. Do I have an answer? Do I recommend something? I recommend that we find out what's the right answer in time and space by patients participating in clinical trials. But I don't think that anybody has a clear answer to say, right now, with a specific type of immunotherapy, if you do that, your patient survival will be improved with X percentage.
Just a second part of that question, Dr. Bota, thank you for that. So on a patient with an immunotherapy, let's say it matched perfectly the DNA and everything of the tumor, and on that secondary round, is that possible to go into second trials after you've gone through a first trial? In other words, are you excluded usually in percentage forms if you do a clinical trial so that you would be possible candidates?
So in general, for rare diseases, so the question was, if you do for a brain cancer an immunotherapy study and you are failing that immunotherapy study, at that time, can you go on a different immunotherapy study? And again, in general, the answer is, at the time of recurrence, you'll qualify for a different study, but every company and every investigator might design their studies in different ways. There are going to be inclusion and exclusion criteria, and I could see if both modalities go onto the same immunotherapy mechanism, that one study might exclude from the other, but in general, no. Go ahead, sir.
Yeah, I have another question about the optic nerve. I've heard that the optic nerve, for those of us who suffer from glaucoma, I've heard that the optic nerve cannot grow back. I've heard many times it's like teeth, it's like hair, or once you lose your optic nerve, there's nothing we can do to get it back. Do you think the stem cells in the future will be able to regenerate the optic nerve? What's our chance?
so the question is first pointing out that the optic nerve does not grow back, and then the question is, what role might stem cells play in optic nerve regeneration in the future? First off, I'll just agree that the optic nerve in a human does not spontaneously regrow. Interestingly, it can regrow in a goldfish and a lot of lower vertebrates like this, okay? So some salamanders and whatnot, they can literally regrow an optic nerve, and it works. So that's something they have on us. So That's a challenge. So when I say that our cells might work in glaucoma, I'm not saying they're going to regenerate an optic nerve. What I'm suggesting is this neuroprotective strategy could help preserve and revitalize what's still there. And I think that's a benefit anytime you have a progressive condition that just keeps progressing. You want to do something to turn it around the other way, even if you're not going to get back to baseline. Now, in the future, it may be possible to regenerate the optic nerve. Remember, in a goldfish, the distances involved are much smaller, so that's a challenge for us. Also, the visual system in the human, it comes back from each eye, it goes to this chiasm, and then things get really complicated and things get rearranged, and some of them cross and some of them don't cross. And then they all have to end up in the right place and form a topographic representation of the visual space back there at the level of the brainstem. Not saying it can't happen, because it works in goldfish, okay? So it can happen. I think it's a possibility, but it's not the easiest one. In other words, that's why we're doing what we're doing with the intent of getting into people, help them to the best we can with the tools available now, and we need a new generation of people to pick up this idea of regenerating the optic nerve in an adult human.
A question about your RP trials. In a typical trial, it might be blinded so that the patients may or may not know whether they're getting the active drug or device. The untreated group, is there anything that is injected into the vitreous? The cells are put in to the untreated group, but
Yeah.
untreated,
So the question is, what's going on in the untreated group? That's going to vary from trial to trial, but in our trial, we do what we call a mock injection. So that's how you keep as many people as possible uninformed about what actually happened. So there's a syringe prepared, comes in the same box, everything's the same. They're prepped in all the right way. But we don't actually inject in the eye. That would be what we call a sham injection. You could inject a solution there, but
You'd know, though, wouldn't you?
huh?
The patient would know whether you're.
Well, it's how many times have you gone through this, for instance? You see what I mean? And there are numbing drops, but I understand what you're saying, and the FDA is concerned that the patients will figure it out, because patients are pretty crafty that way. And that's why we actually have another factor built into this, and we have 2 different dose levels, because nobody knows what dose they're getting. And so we want to, if we can see evidence of a dose response, that's additional evidence. So yes, from a scientific standpoint, to be a purist as a scientist, you're going to say, let's inject all those other people with saline just to be sure. But from an ethical standpoint, I have big issues with that, because the risk of saline injection is, in my humble estimation, at least 75% the risk of the whole treatment, maybe more. So in other words, you're putting them at equivalent risk, and you got to have a pretty good rationale for that.
Found
yeah.
that the use of stem cells have helped the macula.
Have the stem cells help the macula? Okay, that's a loaded question. Yes, they have helped the macula, because our patients would not be able to see as well as they have based on the data unless there was improvements in macular function. So I call that helping the macula. But the undercurrent to what you're saying is, does this help in macular degeneration? And I can't answer that because we haven't actually tested it. But the idea of helping a macula certainly gives us reason for optimism, but it's not the same as testing in a patient with that particular condition.
How about slowing down the blood cells in the choroid layer?
Oh, so what about the choroid layer? So a little background for everybody. There's this complication in a certain form of macular degeneration where the choroid gives rise to abnormal blood vessels. And these abnormal blood vessels can be quite damaging to the retina because they can leak and they can distort. Think of the retina like a film and a camera, for anybody who remembers film and a camera. If you rumple that film, you distort the image, right? So it has to lie flat to give you a good image. So if you have these abnormal jumble of blood vessels, it can literally distort that retina and make for jumbled vision. But if it leaks, the blood can literally kill the photoreceptors and cause all kinds of problems. So it's a bad thing to have these abnormal blood vessels, and when that happens, now there is a treatment for that particular complication, and so that's the Lucentis or Avastin that you hear about out there that doctors are giving. Finally, there's some kind of treatment for macular degeneration. It's not really treating the macula, it's getting rid of those abnormal blood vessels, and it's very good at doing that. But a lot of people, eventually, okay, it got better and then it didn't really help because you haven't gotten rid of the macular degeneration problem, but you at least got rid of these very unruly blood vessels.
So would your procedure be better?
Oh, yeah, I guess I should relate it to mine. Ours, so when I talk about glaucoma or AMD, wet AMD like this, I'm not necessarily saying our cells come in and kick out the treatments that's already there. I'm just saying where there's an unmet need. So in macular degeneration where you can treat those blood vessels, but you still have the degeneration problem, maybe our cells can help with that part of it. You see what I mean? So I'm thinking of multiple ways of treating it. In glaucoma, you have this pressure problem in the front of the eye, and they have drugs that can treat that very well. But then there's the problem with the nerve degenerating. So you see what I mean? I'm not saying we're going to stop doing the drops. That takes care of the front of the eye. We can maybe help with the back of the eye. That's the strategy. That's the way I'm thinking about it.
I think we have a question here.
Macular holes. We've made 3 efforts to close a macular hole in my right eye. And repeatedly we found that I did not generate the cells necessary to close. They started generating and then they just stopped. My ophthalmologist has said that ultimately it's probably going to be stem cell therapy that is going to provide an answer. Has any of your research focused on macular holes? There's no disease.
Yeah.
Find nothing.
So the question is, what about macular hole? So this is a condition where the macula, which of course is the central vision, develops a hole right in the middle. And the fact is that the macula is relatively thick compared to the rest of the retina, except in the very center. That fovea is actually thinner. That's the thinnest part of the retina. And so if there's any kind of traction on the retina, you can develop a hole there. It can just kind of rip open. And so when that happens, obviously you're losing vision right in the center. The question is, would our cells help? And this isn't the first time I've been asked this. But it's really hard to answer. Partly it relates to what you said. It's hard for us to study in the lab because our animal models don't model macular hole. So our animals don't have maculas, nor do they have holes. So we'd have to either get a monkey or something and give it a hole in the macula, or just get a feeling a safety adequate and a good rationale for why to test in humans. Although, you know, I just don't know exactly how it would work. But the key is you're trying to get that hole to just close spontaneously. So obviously the key thing is to get rid of any traction that's pulling it open. And I don't know that our cells would help that. I don't think it would hurt it. The idea for stem cells to fill in the hole isn't that good either. You want to pull the hole shut. The stem cells might do that, the progenitors, they might. But that's just, you know, they might not. It's something you'd literally have to try. And you'd have to get them right in that hole, too.
Questions on that corner before we close.
Oh, right, this will be the last question. Thank you, doctors.
Hi, I'm an RP patient, and I have edema in the back of my eye as well. And I also saw Dr. Boyer and Professor Bird and all those people. And I'm wondering, in your clinical trials, would you accept somebody that has edema as well as RP?
Okay, so the question is, what about macular edema in RP? And so people need to know that RP is photoreceptor degeneration, but there's this added complication that can happen where the central vision suffers because there's accumulation of fluid in the macula, called macular edema. And we've made the decision to exclude that from the current trial. The reason is that macular edema can, to some extent, with some reliability or variable reliability, be treated by other things. In other words, there are pills that you can take for macular edema. And macular edema sometimes just goes away spontaneously. So since right now we're trying to demonstrate efficacy, we don't want the argument that, oh, you know why they see better? It's just their macular edema got better. That's all.
But my
You know.
central
Yeah.
vision is very clear.
Is it? Okay, so there's a nuance to this. If the edema's outside of the macula, then you might be eligible. So it has to be in a place that's not threatening the vision. So that's the other thing is, for all we know, our treatment will help the edema. And we have some animal data from a different model that suggests that it might have some good influences on vascular leakage. That's a big maybe, but we've got some data. So we're interested in testing that, but we have to be very systematic about this.
I guess what I would like to ask you guys all as a conclusion is, if you have questions that are specific to you, we'll be happy to take those as individuals. You have all our contact information. But I'm a regulatory person. I showed you all the whole FDA, so we don't want anybody to close Dr. Klassen or my studies, as this is a public forum. So we will be very happy to take it one by one, taking into account patient privacy and HIPAA regulations. And thank you so much for being here tonight.